Hello class, a very good morning to all of you. Myself Vivek Prakash Yadav and I am here to take your biology class of class 11, and the chapter that we are dealing is photosynthesis. And in our continuous classes, today we will going to be discuss about Calvin's cycle. In the introduction we have discussed that this cycle is divided into two types, that is C3 and C4 cycle. So today we are going to discuss this Calvin cycle, C3 cycle. We found that few plants are having C3 and in few plants this cycle is named as C4. Reason behind that, the first stable product which is formed, the first stable product which is formed is actually if 3 carbon, that is C3 cycle, and if it is 4 carbon, that is C4 cycle. And mostly in desert plants or crystallization plants, that C4 cycle we have found in the family Euphorbiaceae. So it's okay. So in today's class we are going to discuss about Calvin cycle. So this is your class 11 biology class, and chapter that we are discussing is photosynthesis. And in this today we are going to discuss Calvin. So before going to start, see, the Calvin cycle is actually, this is named such on the honor of the person who invented this, or who studied this cycle. So it is actually the name of the scientist, who mainly started here or start the work is Melvin Calvin. Melvin Calvin. So in the honor of the, he is actually a biochemist, he is actually a chemist. So in the honor of the chemist, the name, in the honor of the chemist, the name was given as Calvin cycle. Just a minute. So in the honor of the scientist or chemist, the name was given as Melvin Calvin. But actually not only Melvin Calvin, but he has supporters. He has few supporters, means other persons are there also in his group who have played very much important role in the study of this complete pathway of the C3 cycle. So who are the other scientists? Are here Andrew Benson, James Borsan. So these are the three scientists. These are the three scientists who mainly study the complete pathway of C3 cycle. And in the year 1961, they awarded Nobel Prize. They awarded Nobel Prize in chemistry. Because of this study of this complete pathway or invention of this C3 cycles in the plant. Now, so we are going to continue. What happened in Calvin cycle? Actually, this Calvin cycle I told you is the result of diet reaction. The diet reaction. The result of the diet reaction which is taking place during the photosynthesis, means actually complete formation, or we can say the reduction biosynthetic phase, that includes your biosynthetic phase. So where does this cycle takes place? We all know the photosynthesis takes place in the stomata. Photosynthesis takes place in the chloroplast. So in the chloroplast, this C3 cycle that we are discussing, or biosynthetic phase, or the direct reaction, is mainly takes place in the matrix or in the stroma. Takes place in the stroma of the, so we can say Calvin cycle takes place in the stroma or matrix, in the stroma or matrix of the chloro, in the stroma of and matrix of the chloroplast. Now here we are going to first write the complete reaction which is going to take place in Calvin cycle, and then we will going to discuss about that. Okay, so actually in the C3 cycle, okay, 6 carbon dioxide, okay, 6 carbon dioxide, 12 energy molecules of NADPH plus 18 ATP. Okay, 18 ATP. Okay, how many? 6 molecules of carbon dioxide, 12 NADPH and 18 ATP. Okay, required. Now when these are the raw material, we can say, yes. Now one more thing, there are near about 10 enzymes. Okay, there are near about 10 enzymes which is going to be participate in this complete cycle, because there are near about 10 steps, and each step there is participation of particular kind of enzymes. Okay, so what will happen? There is presence of enzyme. Okay, there is presence of enzyme. Now what you get here? What you get here is your first. This cycle is actually taking place for the formation of glucose C6H12O6. Okay, plus you will get water molecules. Okay, 6 water molecules, and the energy molecules which I use, this is going to be 12 NADP plus 18 ADP, because when they are going to be break down by the reaction, you will get 18 inorganic phosphate. Okay, so this is the complete reaction. This is the complete reaction of the Calvin cycle. Here you can see one thing. When we are going to be discussed in detail, I will tell you how all these things come. But remember, how many carbon dioxide required? 6. Okay, how many water molecules produce? 6. What is the total number of NADPH? That is 12. Okay, so after using, they are going to be break, energy molecules going to be break, and how many? 12 NADP plus release. ATP, 18 used during the process. Okay, during the process, and outcome is your 18 ADP, and we all know that ATP is actually triphosphate bond. Yes. So when this is going to be used, ADP and inorganic phosphate, 18. So this is the complete. And in the biosynthetic phase, or in the C3 cycle, how glucose takes formation or the carbohydrate reduction takes place, that is the important thing. So you will get here what? Glucose molecule. You will get one glucose molecules on the completion of complete C3 cycle. Okay, I mean to say, see, when 6 times this cycle is going to be participate, then you will get one glucose molecule. You will get one glucose molecule. Now the thing now we are going to be discussed. I told you that name of the discoverer who worked on this when they got Nobel Prize, what were the others, and I told you about this, that Calvin cycle takes place in the stroma or matrix of the chloroplast, the complete reaction of the photosynthesis. Now, the initiation of the process of photosynthesis, how start? Okay, we will going to discuss there. Okay, just a minute. Now, second thing that need to be considered or we need to be careful is that I told you, actually there is biosynthetic means conversion of the carbon dioxide or reduction of the carbon dioxide to carbohydrate. So, here the consumption of carbon dioxide or absorption of carbon dioxide is going to be takes place. So, what we need to be remember here, who is going to accept that carbon dioxide or what is the name of, so RUBP, you can say ribulase 1,5 this phosphate, the name is given as, this RUBP name is given as ribulose 1,5 this phosphate. Okay, this is actually the carbon dioxide acceptors. Okay, and this is the molecules which is present abundantly in the biome, means in the earth, because each plant having, for each plant which is undergoing the process of photosynthesis or Calvin cycle, they must have this RUBP as electron acceptor, as carbon dioxide acceptors. All this is also written as RUBP. Okay, that has both are the same thing, just naming pattern is other different. Ribulose 1,5 diphosphate. Okay, 1,5 diphosphate. So what is it? Is first carbon dioxide acceptor. Got it? Carbon dioxide acceptor. So who is the carbon dioxide acceptor here? That is ribulose. Okay. Now the second thing, once this RUBP, okay, once this RUBP going to accept the carbon dioxide, okay, going to accept the carbon dioxide, then what will be the first product? What will be the first product? Now remember here, actually this Calvin cycle divided into two types, that is C3 and C4. Depending on this, that when RUBP, that is ribulase, is going to accept the carbon dioxide, what will be the first stable product? So here you will get in C3 cycle the first stable product is 3-PGA, phosphoglyceraldehyde. 3-phosphoglyceraldehyde. Okay, this is the first stable product. Now what we will get, actually two molecules. Okay, two molecules of 3-PGA. Okay, in one completion. Okay, two molecules of this 3-PGA. Okay, phosphoglyceric acid, you can say phosphoglyceric acid. Phosphoglyceric acid. Okay, this is the first stable product in case of C3 cycle. Now, so what is the acceptor and what is the first stable product? Actually, the complete process of photosynthesis cycles or Calvin cycles, okay, takes place or is actually differentiated into 3 steps so that it can be easily understood. Okay. still we can say this unit or divided into three different stages so that we can easily understand the cycle. What is the first stage, then second stage and third stage? So in the very first stage, what happens? Simply the absorption of carbon dioxide, yes, by the RBP. So that is known as carboxylation. Carboxylation. In the second, what happens? This is also a step of reaction in carboxylation. Carbon dioxide going to be trapped by RBP, then it is going to be converted to PGA, 3-phosphoglyceric acid, and further on, this PGA is act as intermediate product, and that is finally going to be reduced to glucose, starch, or carbohydrate, whatever the product actually. Okay, glucose that we have actually known here. So what happens? That PGA is after carboxylation, the intermediate product of carboxylation going to be reduced to reduction. Okay, reduction reaction takes place. Now after reduction reaction, your carbon dioxide, after reduction reaction, your glucose is formed or the carbohydrate is sucrose is formed. Now what next? As we are using the term cycle, yes, if we are talking cycle, it means that from the point where it is started, okay, from the point where it is started, it is need to be finished also at the same place. Means it is started from the RUBP. Yes, ribulose 1,5 bisphosphate. Ribulose 1,5 bisphosphate is actually initially started and act as a what? Carbon dioxide acceptor. Correct? Carbon dioxide acceptor. As a result, what will happen? First level product is 3-PGA, phosphoglyceric acid. Yes, this PGA undergoes the process of reduction. Yes, undergoes the process of reduction. Okay. Undergoes the process of reduction, and during the reduction, your what? Glucose. Yes, yeah, or sucrose. Your product means actually required product is formed, and after formation of these again need to be reformed. Then only cycle will be continued. That is regeneration. Regeneration. Okay. Regeneration of this RUBP takes place, and this one is your carboxylation. This one is your carboxylation. Okay. So accept. We can say after this point we will come here. Okay, that RUBP going to be accept this carbon dioxide. The stage is known as carboxylation, and the reaction takes place with the help of different enzymes, and the product form, intermediate product form, that is 3-PGA. This PGA is undergoes the process of reduction, which is multiple step process. Okay, multiple step process. After reduction, with the help of different enzymes, it is going to form your desired product. Means whether the food that actually they are forming, that is glucose and starch or carbohydrate. Okay, so this is from now. After formation of this, the RUBP need to be regenerated in the cycle so that the cycle can be continuously takes place. So what happened here? Regeneration steps takes place, and your RUBP again forms, which again going to be continued with the cycle. So this is simple steps, 3 steps what is taking place during the process of C3 cycle or Calvin cycle. Now the thing that we need to be notice here in the reaction, if you remember initially I have mentioned the reaction, there is involvement of 12 Na, GPH and 18 ATP. How do I recognize this? Now listen. At a time there is single molecules of carbon dioxide going to be accepted in each cycle. So how many molecules we have taken there in the reaction? 6. So 6 times this cycle will run. 6 times this cycle run to produce one glucose. In 6 times acceptance of the carbon dioxide going to give you one glucose. So during this 6 times if you remember the cyclic and non-cyclic photophosphorylation that we have discussed, so in that we have discussed that is in non-cyclic form 2 and in the cyclic form 1. That is total how many? 3. How many? 3 ATP is used. Okay, 3 ATP. So 3 ATP going to be multiplied. Okay, 3 ATP going to be multiplied by 6 times. That will be become how many? That will be become 18. Yes, 6 ATP molecules. You know the source. Okay, sorry, 3 ATP molecules. 3 ATP molecules going to participate 6 times. Yes, equal to 18. So in this way, actually what happened in the regeneration phase? Okay, in the regeneration phase, 1 ATP required. Okay, after glucose, the release of this RUBP needs 1 energy. Okay, dephosphorylation required. 1 ATP here and 2 ATP required during the reduction of the 3 PGA to the glucose. Okay, so 2 plus 1 total 3, and here required 2 NADPH. NADPH. Okay, how many? 2 NADPH energy molecules. So this cycle is going to be run how many times? 6 times. 6 carbon dioxide molecule going to be accepted by RUBP, then 1 glucose is formed. So simply 6 cycle into 3 ATP become 18, and your 2 NADPH into 6 equal to 12. So 12 NADP and 18 ATP are used in a complete C3 cycle for 1 glucose molecule, and byproduct this 18 ATP and 18 inorganic phosphate and 18 NADP plus molecules. Okay, so this is what a complete C3 cycles we can say. This is how this complete C3 cycle going to be takes place. Hope you people understood. There are different types of enzymes are there. See, in the case of Ribusco, I told you this molecules, okay, this proteinaceous compound going to accept carbon dioxide in the presence of an enzyme, that is known as this is going to be takes place in the presence of an enzyme that is ribulose bisphosphate decarboxylase. Okay, it is named as the enzyme which is going to be help in the acceptance of carbon dioxide is ribulose bisphosphatase. Now this CO actually it is carboxylation plus oxygenation. Okay, so ribulase bisphosphatase carboxylase oxygenase, carboxylase oxygenase. Okay, so this is the enzyme which helps the Ribusco to accept carbon dioxide. So hope you people understood the concept of C3 cycle, and just if you people have any doubt, you people can leave a comment in the comment section. See, remember this in the regeneration stage there is one use of 1 ATP to release this RUBP. Okay, thank you and have a nice day.